Hello everyone. What we're going to talk about in this lecture is macroevolution, that branch of evolution that ultimately leads to dramatic changes in living organisms, which can lead to the formation of completely new species. So, what we'll talk about in this lecture, first of all, we'll define what species are. Okay, this is the process that might lead to the formation of species, but what are species? Then we'll talk about the importance of the concept of reproductive isolation in the definition of species. Then we'll talk about how species maybe vary throughout different geographic environments. We'll define also the concept of subspecies related to the geographic variation. And then we'll talk about the mechanism of speciation. How are new species formed? We'll talk about the geographic mechanism and the genetic mechanism of speciation and how they are interconnected with each other. So first of all, let's define what a species is. There are different ways of defining species. We'll go through that in the next few slides. In general, we can summarize a species as a population of organisms that are capable of interbreeding, so bred with each other, and produce fertile offsprings. Both of these components are important to define a species. There are species that indeed can interbreed but cannot produce fertile offspring. So a species to be defined as such, or different population to be put together as a single species, you must have this population be able to interbreed with each other and produce fertile offspring that can carry on the genetic pool. Now, this definition, as you can see, obviously applies very well for sexual organisms. So where you have indeed different organisms mixing with each other. It doesn't go well with a sexual organism such as bacteria. But we can see there are different definitions that can be used also to define asexual organisms. Now, the science, very few words about them because we use the classic nomenclature dictated by taxonomy. So, few words about taxonomy. We'll talk about more in the next lecture. That's the science that basically has the goal of grouping organisms together, defining ultimately their identities through by grouping them first in very large groups and then progressively in smaller and smaller group until they can be clustered together in a single group defined as species. Now, species in general, they are always identified using a binomial nomenclature. So a name that consists of two components. The first one is actually the genus. For example, Homo. You see an example here of how taxonomy has clustered humans starting from large groups, so we're all animals, believe it or not, belonging to the Chordata phylum and so on. And then the species identified, our own species, with two names. So the first one corresponds to the genus. There are different species belonging to the genus Homo, like Homo neanderthalensis, Homo habilis, and so on. We are Homo sapiens. And then the second one is what's referred to as the specific epithet. That's the one that really defines the species. But when you define or you refer to our own species, you always use both names, Homo sapiens. Same thing for other organisms like the bears. You have different type of bears belonging to the genus Horsus. You have the Horsus maritimus, which is the polar bear, and Ursus arctos, which is the brown bear in them. Now, species, so we went through the generic definition of a species. In reality, there are different ways that can be defined. Organisms that can be grouped into a species. So you can use what is called the morphological species concept. You can use the biological species concept, or you can use the phylogenetic species concept. Let's go through each one of them. So when you use each one of them, you see, first of all, that they all have an advantage and some disadvantages that come with that. So indeed, in the end, species are always defined as a combination of all of this. So morphological species concept basically uses the anatomical characteristic of organism to group them in specific species. Now, this is great because it can be used, for example, in fields. For example, if you do bird watching, I love doing bird watching. I'm not an expert, but I love looking at birds. These are, for example, two species here that are found, woodpeckers that are found in Toronto as well. You can find them pretty much anywhere if you carefully look around. And They look very similar. They look like actually the same type of animal, but in reality there are two different species. They are referred this to the the downy woodpeckers, and these are the hairy woodpeckers. Very similar, they have the same red dot on the head. But if you look carefully, you can actually identify them as different species morphologically because of the two main differences. One is the body size. The hairy woodpecker will actually be bigger than the downy one. And the bill size. The hairy woodpecker has a bill size which is pretty much the same length of the head, whereas the downy woodpecker actually has a shorter one, half to 1/3 of the overall head. There are other differences like sound and so on that can be used, but these are the main morphological anatomical characteristics which makes them group into two different species. So this is the morphological way of defining a species. This is great because it can be applied not only to sexual organism but asexual organism as well. Just by looking at bacteria you can see how the differences between them. Obviously this is impractical though when it comes to species that have a very large variability, such for example these snails right here. Indeed, these are all part of the same species, but based on the anatomical characteristics you will be brought to believe that there are actually different species. The second way of defining species is referred to as the biological species concept, where species are defined as a group of interbreeding natural populations, which we used before as well, which are reproductively isolated, so they cannot interbreed with other populations. That's indeed the definition we used before. Now, this is great because basically it somehow stresses the fact of the genetic similarity between the population belonging to the same species, and also how different they are compared to other species. So it stresses on that. However, again, cannot be applied to a sexual organism because it refers to breeding, and this takes place only in sexual organism, and obviously it cannot be really be used when it comes to fossils. You don't really have information about the capacity to interbreed or not between different fossils that you can find. So it has its own limitation. The third way to define species is slightly more, it's an ancient one but it's been modernized now with the new molecular data analysis capacities that we have, and is based on is referred to first of all as the phylogenetic species concept, and basically uses morphology, so the anatomy of organism and the genetic sequence data to group populations within specific species. And now they use that also to group them and make this grouping, visualize this group through evolutionary trees, which we'll talk about in the next lectures. The big advantage of the phylogenetic species concept is that indeed can be applied to any living organisms, whether they're also living now or they've been living in the past. So it can be used for fossils if you can actually extract some DNA, or you can actually use that, analyze that, and group these organisms in a much more accurate way than if you were just using the anatomy or you were using the biological species concept. And it can be applied also obviously to sexual but also asexual organisms, since you can use, for example, the DNA. You can extract it from all of them, compare it, and group them together. But the definition that we'll use here of species is a group of population that can interbreed, successfully interbreed, and therefore produce fertile offsprings. So moving on to the next topic is we define species, and we see how the reproductive component, the reproductive character of a species is key to decide whether some organism can be grouped together or not in the same species. So how is this achieved? Well, different species cannot interbreed. So there are we say that they are reproductively isolated. So reproductive isolation is central to the definition of the species. How is this achieved? This doesn't mean that different species cannot interbreed. Okay, as I said before, different species can, or member of the same species can interbreed with each other and be fertile. So both components have to be present. So some species can actually interbreed but not being fertile, and therefore are separate species. So now, how is this overall reproductive isolation achieved? This is achieved through different mechanisms which are referred to as reproductive isolating mechanisms. And now, these are grouped into two different categories based on the fact of whether they take place before the formation of a zygote. So you may have species simply that don't mix together, they never give origin to the formation of a zygote, and therefore they basically use prezygotic isolating mechanisms. Or there are species that do actually can mix together, form zygotes, but they have other mechanisms that take place after the formation of the zygote that prevent the formation of fertile offsprings, and these are all grouped together as postzygotic mechanisms. So let's go through these type of mechanisms. First of all, the prezygotic isolating mechanisms. Now, some species, again, they can interbreed, but they cannot produce hybrid offsprings. So now, in terms of prezygotic isolating mechanism, there are 5 different types. There are 3 here and 2 in the next slide. And so it depends, they're defined as they do take place. But all of them, they prevent the formation of a zygote. So the first mechanism is referred to as the ecological prezygotic isolating mechanism. And basically this applies to species that they actually simply do not form viable zygotes and fertile zygotes simply because they live in different regions. For example, if you take lions and tigers, you can actually put them together in captivity and they could actually produce fertile offsprings. However, they do live in different habitats. Lions are found in grassland, tigers they live in forest in India. Both of them live in India but in different regions, or would be in different areas anyways. And because of that, simply they never meet, they do not form viable and fertile offsprings. So this is an example of ecological prezygotic isolating mechanism. Another one is the temporal isolating mechanism. In this case, these species can actually live in the same place, same geographical area. The difference though is when they mate. For example, if you take the Drosophila persimilis and Drosophila pseudoobscura, you don't need to remember these names, they could actually interbreed and they could produce fertile offsprings. However, the difference is that these species of Drosophila actually mate in the morning, whereas this one only in the afternoon. So they are active at different time and therefore they never interbreed, they never produce fertile offsprings. So that's a temporal isolating mechanism. And then you have a mechanical prezygotic isolating mechanism, very direct. Species could actually produce fertile offspring, but mechanically they can't do that. One example is in these snails, Bradybena. Now these snails, they look identical to each other. However, if you look carefully, these are actually 2 different species, and the difference between them is in the orientation of the shell. So one is Dexter, the other one is Levis. And because of this difference, now the genitalia of these 2 different organisms here cannot connect with each other. Mechanically, they could produce fertile offsprings, but mechanically, they can't do that. Okay, and so this is referred to as a mechanical isolating mechanism, or prezygotic isolating mechanism. The 4th type of prezygotic isolating mechanism, which prevents the formation or the interbreeding, is referred to as the gametic isolation. So some species, they can do it, they mate at the same time, they live in the same place. However, when the sperm and the egg meet with each other, simply the receptors that allows them to connect with each other and fuse with each other, transferring the genetic material, they don't match. So there are differences there preventing the fusion of the sperm with the egg. And one example is what you have here in sea urchins. That's a common mechanism of gametic isolation. The last type is what we refer to as the behavioral isolating mechanism. So in this case, this applies to species that live on the same place they mate at the same time, they could actually have fusion of sperm and egg and so on. However, their mating behavior is different, so that the opposite sex do not get attracted to each other. The example you see here is the one that takes place in fireflies. Fireflies, there is the male, to attract the female, it does a series of flashing. Okay, there is a specific pattern that they use. Now, in different species, here you can see 4 of them, but in reality there are more species, and they all have, the males, they all have their own specific pattern of flashing that they use to attract the female belonging to the same species. For example, if you look at this one right here, the male will actually do that first of all at a fairly high distance from the ground, and then it would have this regular flashing, brief flashing here. So it would flash, go dark, flash again, and so on, and it will move at this level. Now, these species here have a similar pattern, but is done first of all closer to the ground, and when it flashes, the flashing time is longer than these other species. So this is not recognized by the female belonging to this species here, but only to the female belonging to this. And then also, other than the distance, so the way in which the male moves contributes to. So is the pattern, the behavior of the male, ultimately, that prevents female from species that theoretically could actually interbreed to be attracted by that specific male. And so this keeps the separation between the different species. So as you can see, the capacity to interbreed is not written in stone. It could happen, and in captivity sometimes you can even force it. You could even produce new species. But in nature, all of these mechanisms to naturally prevent this from happening. So these were all the 5 prezygotic isolated mechanisms. Let's look at the postzygotic. So in the postzygotic mechanism, instead what happens is that indeed member of 2 different species can actually interbreed. However, the zygote that is formed has limited fitness, so viability, or fertility. So in the long run will cause the death of the new lineage that could be formed. So there are 3 different mechanisms here. So the first one is referred to as the hybrid and viability postzygotic isolated mechanism, where you have simply the hybrid that is formed. So once the sperm and the egg fuse together, the hybrid that forms, it dies either in the embryo or a very early stage. We have an example. For example, if you mix a goat and a sheep, they actually can interbreed. However, the result of the fertilization is stillborn. So the baby is born after the gestation dead. Okay. So again, there is fusion, but the hybrid is not viable. The second mechanism is referred to as hybrid sterility. So in this case, the fusion of the egg and the sperms leads to the formation of a new organism. However, this organism is sterile. And an example is what you get when you mate a horse with a donkey, producing what is called a mule. Now, there is a difference in number of chromosomes in horses versus donkeys. And these ultimately, so you have 32 pairs in horses and 31 in donkey. So when the 2 gametes mix together, now you end up having basically a mule that has 62 chromosomes plus 1 unmatched one. And this is what causes the sterility ultimately of the mule. So even though you're producing an organism through interbreeding between 2 different species, now this new organism would not be able to carry on. Okay, so there is going to be sterile. The last postzygotic mechanism is what is referred to as the hybrid breakdown mechanism, where you have that indeed, in this case, the fusion of the sperm and the egg leads to the formation of a new organism that can actually survive, can actually be fertile for the first generation, but then in the long run leads to the formation of a second generation that it's either as reduced survival or reduced fertility. An example is what happens in Drosophila. So here you have, if you cross, for example, Drosophila melanogaster with Drosophila simulans now here, these 2 organisms, they're very similar. They can interbreed. However, there are two genes. This is one of the possible explanations, but the most commonly accepted one is that they actually carry two genes which normally they are separated with each other. Okay, so this is not a problem for all members belonging to the same species. This is a different gene with different alleles. But however, when you mix them together, the combination of the alleles of these two different genes causes male unavailability, so the male will die, and sterility in the female, so the inability to produce further generation. So reproductive isolation is key to define species, and it's achieved either through prezygotic or postzygotic isolating mechanism. Now let's look at how species are formed. How are new species formed? In order to understand that, though, we have to understand also how is the same species can be variable in different geographical locations, and how this variability is spread to different geographical variation. Understanding that will make us understand later on also the geographical mechanism that can lead to speciation. So geographical variation play a key role in the variability of organism. So if you take one species living all in the same area, very close area, will be very similar to each other. There will be very little variation. However, many species are spread in the earth through different geographical areas that can be connected with each other. And this has led to the same species to accumulate different geographical variation, okay, to the point that you can have population within the same species in different geographical location which can still interbreed, produce viable organism, but they can be fairly different from each other. And this is what you define as subspecies. So these are local variants of the same species that you can find in different geographical location. Now, subspecies that can interbreed, that can produce fertile offsprings, and therefore they can exchange the genetic information, their own variability. So let's go through some example here. The first one is the rat snake in North America. They tend to live in the central part and on the east coast. And here you have that even though these are all members of the same species, you can have fairly different looking type of rat snakes. So if you look in the north, you will find mostly black rat snakes. They're all dark in color. That's what you can find in Canada as well, by the way. Here you can find on the east coast, southern or central southern east coast, you can find yellow rat snake, here Everglades, and so on. So these are all different variants of the same species, and they're referred to as subspecies. But again, they can all interbreed with each other, producing fertile offspring, so they are all members of the same species. Similarly, that's what you have with tigers in Asia, or you used to have with tiger in Asia. This is actually a great example because it shows you how ultimately the variation, geographical variation of the same species ultimately can lead in the long run even to the formation of different species. So this is the distribution of the tigers, how it used to be in Asia. You can see particularly in these in the southern part, so in India, in what is referred to as Indochina here, and China, how you had all these tigers basically in continuity except for the one in the Caspian region here, but you can see how there was continuity probably in the past. So these tigers, so the Siberian tiger here, the South China tiger, Indochina tiger, and some extincted one like the Javan tiger, the Bali tiger here, Sumatran tiger, and so on, they're all members ultimately of the same species, even though they have accumulated fairly unique characteristic with each other, but they can still interbreed. They're still considered part of the same species, and therefore they are considered as subspecies here. Now, the distribution of this variation can be different depending. You can have a linear distribution or you can have a circular one. Now, in general, so this distribution can give origin to variation of different types. You can have more distinct variation, almost fairly blunt groups formed, or you can have more even distribution. When you have an even distribution, what you refer to is a clinal variation, where you have small, smooth variation taking place one after the other. You can see the continuity in the population. And now, a specific type of clinal variation is what you find in what we call ring species. We'll talk about that in a second. So when we talk about clines or clinal variation, you're talking about a pattern of very smooth variation which take place through a geographical gradient. That's what is taking place in humans as well. I'll tell you in a second. So now this can be linear or circular. If it's circular, you refer to as ring species. You see an example here. What we mean by cline is the variation you find, for example, in height in this plant, which is Achillea millefolium. You don't need to remember the name. In this region here of the central or west coast, sorry, of the United States. So where you have the Great Basin is this area which is east of Sierra Nevada, and then there is this area right on the ocean corresponding to where California is. So now you can see that these pieces of Achillea millefolium here, if you go to the Great Basin, they tend to be tall, and then progressively going higher up in Sierra Nevada, in the mountains here, in the range here, you see that they get smaller and smaller, and then progressively they get taller again while you get closer to the west coast in California there. So this variation is very smooth and overall linear, is referred to as a cline. Similar thing you see in humans, and that's a big argument against the concept of race. We tend to see humans as races. Races don't really exist. We talk about Asian, we talk about Caucasian. In reality, it's a kind of joke. It's the remnant of the concept of eugenetics that was used in the early 1900s. But in reality, races don't really exist. We are actually clines. There's a clinal variation in humans. If you go from Asia all the way to Europe and even North America, there is a continuous variation. And you can see it in the people that live in between the areas. There is no clear-cut groups of people which will correspond to races, but there is clines. All the variation are smooth in between the different groups, ethnic groups, and so race doesn't really exist. So now, these smooth variations, so this cline can be linear or circular. When they are circular, you refer to the species that have this clinal variation as ring species. So ring species correspond to the geographical distribution of subspecies which are organized in a ring. And so now when they are organized in a ring, and keep in mind this ending can even be closer to each other, even in contact with each other. So what you have here is that these members or these subspecies are organized such that species that are close to each other, so adjacent subspecies like A and B, B and C, C and D, D and E, can actually interbreed with each other and exchange. So there are variation, but they can exchange. They can still mix their genetic pools with each other. However, the last and the first, so the first and the last pieces, so the two end population, they may have so many, they may have accumulated so many different variations that even if they are in the same environment, might not be able to interact and interbreed with each other. So theoretically, A and E could be considered different species. However, they're still considered as subspecies because they're still in continuity with each other through the other population that they can interbreed with each other. Okay, but you can see here, and this is one of the mechanisms that can lead to speciation. You can see how here, if you start just breaking this continuity, some event breaks this continuity, now you could have the formation of two different species as A compared to E. So how the geographical distribution can contribute ultimately to the formation of different species. So here is an example of a ring species, is the one found in salamander in California. Again, always around Sierra Nevada there, the range there. You can see how there are different species that are organized around the Sierra Nevada range there. None of them live on the top, on the range there. So you can have different subspecies here in continuity with each other, forming a ring structure. So it's a ring species here. And now you can have that indeed, if you take the end species here, they may not be able to interbreed with each other, but again, they're still member of the same species. So there's just subpopulation compared to each other, or subspecies, sorry, compared to each other. The reason being they're still in continuity through the other species that can interbreed with each other. Finally, let's talk about which one are the mechanisms that ultimately lead to the formation of new species. How do you get these new species? Now, there are two types of mechanisms which actually interact with each other. Okay, you see that. Geographic mechanism relies still on genetic mechanism ultimately to lead to that reproductive isolation which is necessary to define a species. So let's start with the geographic mechanism, or the geography of speciation. Now, geography can play a big role in the formation of new species, as we said before, and it can be very direct or indirect. So now, based on the mechanism in which species can be formed, you can define three different mechanisms. There is an allopatric speciation mechanism, where allo means different, a parapatric speciation mechanism, para being beside, species will be, the population will be originally beside each other, then they separate into new species, and then you have a sympatric speciation mechanism, where sym means that they're still together, but some events have led them to lose the capacity to interbreed. Now, all these mechanisms work combined with genetic mechanisms that are either the cause, you see in sympatric speciation, is that genetic mechanism that causes the sympatric speciation, or is generated after this speciation mechanism. You see, when we talk about allopatric and parapatric mechanism, they will lead ultimately to genetic mechanism that cause the inability to interbreed in the long run. And ultimately, all of them, to form new species, they lead to reproductive isolation, whether it's prezygotic or postzygotic, doesn't matter. So let's go through this mechanism. What do we mean by allopatric speciation? Allopatric speciation takes place when you have the member of a population, somehow they get isolated. And these can be caused by natural or man-made mechanisms, like hurricanes or the formation of the Isthmus of Panama, for example. And so which artificially they separate a population. You can see right here. So you have, in the first stage, you have that the populations, so all these members here, this population, they can interbreed with each other, right? Member of the same species. But then there is an event here, formation of a glacier or in isthmus, whatever, that is forming there, separates the organism. At the beginning, this organism can still interbreed with each other, but they don't have geographical contact. They're being separated geographically. Now, if this persists for a long period of time, now this population will start acquiring genetic differences, right, through mutations. You can have genetic drift, you can have the founder effect itself, you may have that the alleles frequencies were different in these two groups somehow. There were two subspecies, so there were some genetic differences, and these are enhanced over time, and to a point that even if the separating feature, like this isthmus in this case, disappears, and now this population can get in touch with each other again, these genetic mechanisms that have been acquired over time, they have made them unable to interbreed or produce fertile offsprings, and therefore that made them separate species. Now, this is an example of how the founder effect will work. So in this case, you could consider, for example, this population to be a founder population as being separated from the original population. And obviously natural selection here will cause these organisms or these to adapt differently to the different environments. So it would work both ways. But ultimately, in this case as well, through allopatric speciation, what you have is that the separation of these population into two parts through natural selection and founder effect ultimately has caused the formation of two populations that cannot interbreed with each other anymore, and therefore two different species. Now, this separation can take place through these geographical events, natural or man-made, or can be generated through migration. So allopathic speciation can take place also because of migration. In this case, obviously, what will cause the speciation will be mostly the founder effect. So the different organisms from the same population, for example, in this case, that migrate to different islands, could have slightly different genetic frequencies, okay, which in the long run, if there is separation there for a long period of time, will still lead to the incapacity of these new population to interbreed with each other, right? If they acquire enough genetic differences, they can't interbreed with each other anymore, and therefore could be defined as different species. The second mechanism is what is referred to as a parapathic speciation. In this case, what you have is that the population belonging to the same species doesn't really get separated geographically, completely geographically, but it gets separated virtually by an environmental discontinuity. One example is what happens to this bent grass belonging to this species across this tenuis. You don't need to remember this name, but you have to remember the mechanism here. What you have is that within the same field, you can have the different environmental condition. Okay, so for example, pollution may cause the formation of first of two different strains. So you may have some strain that adapt very well to a polluted area. For example, in this bent grass, you may have copper that favors a certain strain to grow, but not the other one. Whereas the same strain that grows well in the polluted, copper polluted area here near the mine might not grow well in the unpolluted region. This is causing some separation, which again, in the long run, might lead to the accumulation of genetic differences between these two strains, and in the long run, the incapacity to interbreed between these two species. For example, in bent grass, now these two strains have accumulated genetic differences that makes them actually be fertile at different times. So a prezygotic temporal isolation. So you have that the one in the copper polluted region actually start mating or are fertile, produce flowers 1 week earlier than the one that live in the unpolluted region, and therefore there is this prezygotic temporal isolation between these plants. The last mechanism is referred to as sympatric speciation. In this case, there is no geographical or environmental separation, but the formation of these species takes place because of sudden genetic changes within the population, which lead to the reproductive isolation. One example you have is, for example, this one, moving directly to this, is the apple maggot. These are insects that naturally, until at least 150 years ago, they used to live in other plants. Okay, but 150 years ago, and then they would actually reproduce in cherries. Okay, they will be found to reproduce in cherry. But then around 150 years ago, scientists start noticing that some member of these insects were actually starting mating and reproducing not on cherries but on apples. So now these started like causing a geographical or environmental separation between these two organisms. Not only that, they will be reproducing in different fruit plants and also at different times. How was that caused? Well, so in this case, these two species could actually still interbreed with each other, but now some genetic event has caused them to mate in different ecological spatial area and also at different times. So they have made them accumulate very quickly some temporal and ecological prezygotic isolation mechanism. So that's what sympatric speciation consists on. There is some changes, genetic change has happened within the same population, even though they were living in the same geographical area. There was no environmental difference at the beginning, but some genetic event has caused them in some loci to actually diverge. That's what this picture shows you. This is the normal habitat of this maggot. this insect. You don't need to remember exactly what the type is. So they were laying eggs on cherry trees. But then 150 years ago, they were actually found that some group started laying them not on cherry trees anymore, but on apples. And now, not only that, so they have developed an ecological difference or prezygotic mechanism, so they don't interbreed with each other anymore, but also a temporal mechanism too. So the one that reproduced on the apple, indeed, they reproduce 3-4 weeks before the one that reproduced in the original cherry tree, the other plant there. So these genetic events have caused what is called the sympatric speciation of these organisms. This is for the geographical mechanism, or mostly geographical mechanism, whether they're direct or indirect. So behind all of these, obviously there are genetic mechanisms that have caused the geographic or ecological separation, or vice versa, that were caused by the geographical or ecological or environmental, separation in the long run. What are these genetic mechanisms that ultimately are beyond the formation of new species? So there are different mechanisms there, and they can be grouped in different ways. So there are genetic divergence, which can ultimately be formed between allopatric populations, so populations that ultimately get divided either by migration or events. There is polyploidy that can take place in sympatric populations that exist exactly in the same event. They may actually very quickly, you see, become two different species through a one generation change corresponding to polyploidy. And obviously there are slower mechanisms like chromosome alteration that ultimately can take place in organisms, whether they're living in the same area or they move to different geographical location. They can still contribute to them. Let's look at them. So genetic diversion, these are genetic changes that take place in populations that originally were part of the same population, but they have been separated for a certain period of time. Okay, so these can take several changes. Again, so these were genetically identical populations, but in the long run, being separate, they are subject to different natural selection. Okay, there is the founder effect, but that's only at the beginning. But then the natural selection may be pressuring genetic changes in this population. Okay, changes that can involve different loci. So, and once they involve, for example, in Drosophila, you just take 5 to 10 loci, and now these two species will not be able to interbreed anymore. Just 5 loci, even just 5 loci, or even one. For example, going back, the example of the Bradybaena, if you remember, I used these as a mechanical prezygotic isolating mechanism, where the different orientation of the shell prevents a male and a female of the two different species to interbreed with each other. Well, this change is caused by actually a single mutation. So imagine having all the organism or all the snails being of this type originally. Then some event separates them. All it takes here is one genetic change, okay, just one mutation to cause a change in orientation. Imagine now you have right hand or left hand here shell, one or the other. Now these two populations cannot interbreed with each other. So you can see how quickly the genetic divergence, which is a consequence of the allopatric mechanism of speciation, can cause the loss of reproductive capacity or interbreeding capacity between two populations, and therefore the formation of the species. But you can see how, again, the geographical separation and the genetic divergence are connected to each other to produce, in the long run, two separate species. Another example can be, or happens all the time, this genetic divergence might be also correlated to the sexual selection, where you have males that once they get separated, they accumulate more and more different sexual mate characters, right? Which at a certain point are not recognized by the female of the other population. Imagine this happening in these ducks, right? So originally the male and the males all have the same sexual characters that attract the female. But then the separation causes the male in the two populations to acquire sexual traits different from each other to attract the females. So at a certain point, if you mix them together now because of the sexual selection, the males here don't have those characters that attract the female from the other population and vice versa, so they cannot interbreed with each other. So that's the genetic divergence that can take place, particularly in allopathic populations. Second mechanism is polyploidy, and this can take place in sympatric population. So by polyploidy, what we mean is the sudden change in the total number of chromosomes that you can have in a population. This can take place in one generation, a single generation. Okay, and so once you have that, for example, a diploid population turns into a tetraploid, now you have two population of organisms, one with only two sets, the other one with four, that cannot interbreed with each other because they will form population with three, which are not viable. So how can it take place? Well, there are two ways that this can take place. It can take place within plants of the same species. In this case, you were referred to as autopolyploidy, or members of two closely related species, and that's referred to as allopolyploidy. So when it comes to autopolyploidy, this takes place because of meiotic abnormality that takes place overall fairly frequently in plants. So what you have is that, imagine you have this diploid plant, which through abnormal meiotic event does not separate the members of the homologous chromosomes, and therefore gives origin to a diploid pollen instead of a haploid pollen, and a diploid ovule instead of a haploid ovule. Now, through the process of self-fertilization, you might have that these two pollen, so the pollen, sorry, and the ovule here, these two gametes, they fuse with each other, and at this point they will be forming a tetraploid plant, which cannot hybridize anymore with the original plant it was originated from. So the tetraploid plants now cannot interbreed, with the diploid plant, giving origin in one generation to a completely new species here. So that's referred to as autoploidy because these arise from the same species. You may have similar things happening, in alloploidy. In this case, you may have that the pollen will be coming from one species, the ovule still diploid, both of them, but from a slightly different species. And still they will give origin to a tetraploid hybrid in that case, that will give origin to a new species. Now, alloploidy can also take place within the same species, like in, two different species, not because of meiotic abnormality, but by an unpredictable mitotic event. So what you can have is that fertilization between two closely related species, A and B, take place normally. So they produce haploid gametes, they produce a normal hybrid in that case, right? Which would have only one copy of each chromosome. Normally these will be infertile. However, if you have an unexpected mitotic event where this chromosome, they actually duplicate, at this point they will form, for each chromosome here, now they will form basically homologous chromosomes. They will be identical. They wouldn't be technically homologous, but they are identical chromosomes which now can be separated. So now you can have that meiosis can take place in this plant too, because now you have two members that can be separated through meiosis. In this case, you will not have that. So meiosis can take place, and now through the process of self-fertilization, you can have that this tetraploid zygote, this case, and then plant that's being formed can give origin to another tetraploid zygote that can then carry on in the same way, producing basically a different species compared to the two original one that gave origin to the interspecific hybrid. So in this case, again, it's taking place to two different species, even though they're close to each other, evolutionarily speaking, and that's referred to as allopolyploidy. Now, this is actually a process that commonly used also in agriculture to produce artificial tetraploid plants. And the reason is that tetraploid plants tend to have also higher level, higher dosage of the product of some genes, because you start having, instead of 2 copies, 4 copies, and ultimately tends to produce plants that tend to be stronger to either infection or drought and stuff like that. And so it's very commonly used. The mixing of particularly oligopolyploidy, so the mixing of 2 similar species commonly used in agriculture, again, to try to produce crops that can be more resistant. One example is the bread wheat. That's how it's being produced. So we talked about the genetic divergence, the polyploidy. The last genetic mechanism which ultimately will cause the formation of organisms of different species is chromosome alterations. These take longer. They don't take place usually in one generation. And what do they consist of is different changes that can take place in the chromosomes of a species so that new forms of chromosome can be formed. What are these type of chromosome alteration that can take place? Well, there are different types. There are deletions. So imagine this is an original chromosome in one species. Some chance event may cause the loss on one section in this chromosome. In this case will be B, for example, and therefore the formation of a completely new chromosome which is missing that B section here. So that's referred to as deletion. Opposite, you can have the actual one region gets duplicated. So now you give origin to another new chromosome, which instead of having only one B section would have 2 B sections here. Now in the long run, genetic or selective pressure could actually cause the gene in this section B become different than the one in the original section B here. So even though originally you have the same genes in both sections, now you can have that some of these genes evolve to become other genes. This is what, for example, is supposed to be the origin of the difference between oxytocin and vasopressin. They're both 10 amino acid polypeptides, very similar in structure, but they have 2 different amino acids. And the explanation on how they evolve is exactly through duplication of that gene. The gene has duplicated, it was identical originally, then some mutation have caused the changes in 2 amino acids. Other type of chromosome alteration are inversions, where you have that a full section in a chromosome, so in this example is B, C, D, and E, detach from a chromosome and get reattached in the opposite orientation. That's referred to as inversion. Or what you can have is the translocation, whether it's unidirectional or bidirectional, between 2 chromosomes, that they basically intermix, similar to what happens during crossing over. They intermix, but now at this point you're talking about chromosome, not homologous chromosome, but chromosome of different types. Let's say chromosome 2 and chromosome 7, exchanging material, and these referred to as translocation. Again, all of them cause the formation of new chromosome, which then can be passed on eventually to, if they involve the gametes formation, they could be passed on to the next generation and maybe lead even in the long run to the formation of a new species. Now you can actually visualize these differences between chromosome. For example, if you're looking for translocation, and that's how Down syndrome, for example, can be diagnosed. You can take some cells from the fetus, put them under the microscope and stain them with Jameson. You can use this stain, Jameson stain, and this visualizes a different chromosome. You can see the difference in chromosome by looking at the bands. That's where the stain takes place. So where these Jameson stain, it binds very well, will give you darker bands. It depends on the accessibility of the stain ultimately to the chromosome, so how compact the chromosome is in some areas. And so areas that they have the same bandings are actually the same areas. So if you take, for example, this chromosome from one human and another human, you will see exactly the same banding. Now you can actually compare, for example, the one from different species. So in this case here will be one chromosome taken from a human, one from a chimpanzee, will be very similar to what you see in bonobos as well, which are closely related, gorilla or orangutan. All of these organisms here are members of what we call the hominidae. and we are very close to each other from an evolutionary point of view. And indeed, you can see that if you analyze the chromosomes by James and Staney, you will see that there are so many identical regions to each other, and some differences too. You can show, for example, this is the chromosome that you have here. Now we are the only one having this chromosome, but now if you look carefully, you can see that in other organisms like chimpanzees, gorillas, and orangutans, the same chromosome actually correspond to two different chromosomes, right? If you put these two chromosomes together, you see that you actually get basically all the bands, plus minus some translocations at the end specific to some of these organisms. But you can see how basically this chromosome that we have here may have come from these processes here, chromosome alteration. So you can see how the chromosome alteration ultimately may be involved in the formation of the new species. This was also studied by these scientists, Yanis and Prakash, in the early 1980s, where they have studied actually 1,000 of these bands in hominidae and other apes, and what they have seen is basically all of these 1,000 bands are conserved. They can be distributed in different chromosomes. Indeed, for example, we have 46 chromosomes, where the other primates, they have 48. So, but ultimately, even though they may be organized in different chromosomes, we all have basically the same bands, to a point that if you look at the bands, not at the DNA sequence, okay, you can say that we are basically have all 99% similarities with each other. This tree refers only to hominida, but even with other apes, you can find so much similarities. Again, pointing to how ultimately we might have all originated from some organism here that had certain chromosome and chromosome alteration may have been involved in the formation of the different lineage and ultimately the different species that we might recognize nowadays. So, in summary, what did we talk about in this lecture? We talked about a lot of things. First of all, we talked about the definition of species. So, species, common definition you can use are group of populations, okay, that can interbreed with each other, producing fertile offsprings, okay. Now we went through different variants, if you want, or different ways in which you can recognize different species. So the morphological, the biological, and the phylogenetic definition of species. But when you put them all together, that's the definition that we use. Populations that can interbreed with each other, so it's important that they can interbreed with each other, and, I stress, and produce fertile offspring. So now we talked about also how the reproductive isolation is achieved between different species. So we talked about the 5 pre-zygotic isolation mechanisms and the 3 post-zygotic isolation mechanisms. Then we talked about also how members of the same species that can present different geographic variation, often organized in what we call a cline, so a gradual changes in specific characteristics that you can see either in a linear form or in a circular form when you talk about ring species. And we define also what subspecies are. And finally, we talk about what are the mechanisms, both geographical and genetic mechanisms connected with each other ultimately, that lead to the formation of different species. So we talk about the allopatric, the parapatric, and sympatric mechanism that lead to the formation of a species and how they are connected to genetic divergence, polyploidy, and chromosomal alteration.